Welcome to the Forensic Chat Room. I'm Associate Professor Carl Wiensko.
Professor Roger Bayard.
And in these sessions, we will talk about different forensic topics.
They'll be associated with your lectures, there'll be additional material, we'll be talking to you whether there are topics that you want us to discuss, there'll be interesting cases, and there'll be material attached to it that you can also download. So there's a range of material. Why we're doing this is that we can't really record the lectures as they are because there's some very sensitive material and sensitive casework there, but in this way you can actually tune in, see what we think is important, and we can give you exam tips and talk about life as a forensic pathologist.
All right, so welcome.
Yeah.
Hello everyone, and welcome to today's session where we have Dr. Fiona Bright with us.
Hello.
Welcome.
Thank you.
And we're talking about basically her PhD, but PhDs in general.
Yeah.
Mm-hmm. Great.
And you went through TIFFs with us, didn't you?
I did go through TIFFs. So I did TIFFs in third year of my health science degree, and really enjoyed the lectures and the material, and approached Roger after one of the final lectures to see if I could do an honours project with him and begin some research, because I was really fascinated by the pathology.
I said no, didn't I?
I think initially you might have said, "Oh, come back later," but I persisted. So yeah.
Very good.
Fortunately, yeah.
And you did I wouldn't say a strange project, but you looked at hypothermia, and you compared Sweden with Australia.
Yeah, we found some great findings, I guess, surprising ones, where there were more hypothermic deaths per population in Adelaide, Australia, than in Sweden. And the demographic was older, elderly women who were living by themselves in homes that weren't properly insulated.
And then you decided to move on to the PhD?
Yes, so I had a really great time in the honours and wanted to continue on in the field of pathology with like a bit of a forensic touch, I guess, and studied the neuropathology associated with sudden infant death syndrome, which is the death of an infant under 1 year of age with unknown causes, usually during a sleep period. So we were looking at the brainstem specifically, which is a region that controls breathing and heart rate and the ability to arouse from sleep. And it's a region that we think is, or we know is defective in SIDS infants. So I was looking at that region and different chemicals that control brain function.
Well, that was the point, wasn't it, that the Americans at Harvard had been looking at serotonin.
Yes.
And, we decided to look at substance P.
Mm-hmm.
And so you were in Harvard for 18 months?
Yeah, so I spent the first year of the PhD at Adelaide Uni over here, and that was learning kind of the basic techniques and getting my head around the pathology, and then went over to Harvard in Boston for 18 months to perform some more, some bigger experiments with actual human tissue that was available over there.
Do you remember, do you remember the reaction when you came back with your results? When you put your laptop up?
Yeah, I didn't think I fully appreciated what those results were until I had shared it with yourself and Bob.
Well, Fiona came back and said that she looked at 14 nuclei or areas in the brainstem in SIDS kids compared to controls, and 9 of them no difference, 5 of them significant reduction in substance P in the SIDS cases, which we were pleased with. And then she said that 2 of the nuclei controlled the response to hypoxia, and 3 controlled head and neck movement, which basically answered the question as to why babies who die of SIDS have trouble face down because they can't lift their heads up. So that was pretty spectacular, wasn't it?
It was an incredible finding,
yeah,
and I think that set the basis now for future work to look at specific nuclei and other neurotransmitters that are involved in the brain. So yeah, it was a really great project, and it provided some really amazing outcomes for SIDS research in the future.
Where have you presented that? You've presented
in Norway,
yeah,
and in Amsterdam and Uruguay.
Oh, and so have a good theory of travel.
Yes, the conference attendance was also a great bonus of doing a PhD and travelling with Roger and being able to present to different, colleagues all around the world who are doing the same sort of research.
So how long did your PhD take from start to finish?
Yeah, I think probably 3.5 years, submitting kind of in that 3.5 year time frame. So the first year, I guess, was doing a lot of reading, getting your head around, what are you supposed to do in a PhD, and doing the what are they called, requirements of your first year PhD, so literature review and things like that. And the second year was the data collection and all of your heavy experiments, which was done here and in America. And then the third year was pulling it all together, analysing the data, statistics, and then writing up a thesis.
Yeah, so terrible hard work for sort of 2.5 years, then suddenly something comes out of this
Suddenly
morass.
you have all this data and you're like, what do I do with this? What
Yeah.
does this mean? And you have to pull it together and come up with a really great explanation and story, I guess. And yeah, it's actually very fun at the end. You don't feel it at the time, but looking back, it was those last few months are really exciting to see how much work you'd done and to pull it all together and be able to share it.
Do you think that your honours year prepared you for your PhD?
Yeah, definitely. I still believe that honours is the hardest year because it's 1 year jam-packed of full-on research, and you've come out of undergrad where you've done some group work and you've done some projects on your own, but then you're thrust into this research environment where you really have to be on top of deadlines. And I was fortunate to work with Roger and yourself, and that meant I was able to write manuscripts and be able to publish. And so I was well prepared, I think, going into the PhD, then knowing what was expected of me to be able to write better and see through the research and the gaps and how to pull things together. So
yeah, really is a transition period, isn't it? Going from everything being set up for you and so structured to then up to you.
Absolutely.
Yeah.
So that honours year was really great, and it also gives you the opportunity to build your confidence because you have to stand up and prove how much you know about your topic and defend the work that you've done. And you really can't you need to know your work in and out. You can't get up there and fake it.
Well, you've defended your work at Harvard, so that's pretty good. And so what now? What's going on?
Yeah, so I'm currently doing a postdoc at the University of Sydney. So I'm working in the Department of Neuropathology, and I've transitioned from, SIDS research in the infant brain now to the adult brain, and we're looking at neurodegeneration. So I'm specifically looking at frontotemporal dementia and the different pathological changes in that, and hopefully trying to develop better screening techniques and find a biomarker so that we can have a blood test possibly. So a little bit similar actually to the SIDS research where you're looking for a biomarker before these people pass so that you can then try and intervene in some way or treat them specific to their pathology.
I was just thinking back to the PhD though. I think we were just so lucky that we had you wanting to do the PhD.
Yeah.
We had the substance P work that nobody had really looked at properly. We had Harvard interested, and then we had Rivers Gift who funded you to go there for 18 months.
I think it's kind of unheard of that you have everything aligned like that and you have multiple institutes wanting to collaborate together on the same thing. And we had, the close association with Rivers Gift who were experiencing, they had just lost their child and wanted to fund this research. And then we had, kind of us there being like, well, we want to do this, so, give us this opportunity and we'll make the most of it.
It all
was really great.
All fell in place, didn't it?
And you've had how many papers out in the last?
I think we've had about 5 out in the
just
past
in the last 6 months.
6 months,
So you've cornered the market there.
It's been really great. Yeah, it's been nice to see your work go out and be well received.
So what advice would you have for TIFF students if they're looking for a research career? I mean.
Yeah, well, for me it was just one lecture that I was like, oh my gosh, I think it was actually a lecture about death, pathology and death, but I was just very fascinated by the ability to see different pathologies, at autopsy, but not know that they're there during life. And so it was just that one lecture that kind of inspired me to look deeper and to go, okay, I want to do this. I'm going to go and approach Roger. And I guess just not be afraid to go and approach different people and ask questions.
Yeah.
I remember asking Corinna Vanden Heuvel, how do I be Roger? What do I have to do to be Roger?
She's got too much hair.
That is the scariest question I've ever heard.
I know. I think because you get, as I said, you get inspired by a lecture and then you're like, oh, I want to know how you can get to that point. And as an undergrad, I guess you don't really think, well, how are these lecturers, how have they got to the position that they're in? So asking questions and being
me 300
inquisitive.
years.
Yeah.
And some.
But the health science degree here at Adelaide is just a great smorgasbord of different things that you get to learn. And you learn very quickly what you're interested in and what you're not. And then you can kind of streamline that into being like, well, is university in the long term something for me, or am I going to use this experience to go and do something else in life?
Yeah.
So it was really great. And the PhD I had such a great experience. I know that's not everybody's take on a PhD, but I really had the best experience. So just keep going for what you want, I guess. That's my advice.
That's true.
Yeah, no, it's really
very
good.
good.
Yeah.
Cool.
Is there anything else you want to add that you think that Roger and I should do for the TIFF students to get them more inspired and go on and do honours and maybe a PhD? Should we have is there an approach that you would sort of recommend?
Free breakfasts?
Yeah, that would be great. Anything involving food or beverages, freebies. But no, the TIFS course is already like very enjoyable, and you're learning so much about things you wouldn't even come across, I guess, in life. And I think more just making it known that you can go on to do research, because sometimes that gets lost and people think of it as a backup, but it really is something that is possible to do and to do well and to go on and have a career in.
Yeah, well, I think you're a shining example.
I hope
of that.
so. But yeah, I didn't know that was a possibility. I didn't know about honours or masters or a PhD probably until that third year, where it was like, oh, this is just another option if you want to look into that. But bringing that to the forefront early on, so people who have that knack for wanting to know a little bit more about different things can go, okay, well, I should research more or look more into this.
Well, that's great. And that's why we're doing these video series, so that we can inspire and get you as students to come and see us and do honours and hopefully a PhD in the long run.
Yeah.
Given that there are limited places.
There are limited places.
Yes.
And it's a lot of hard work to do.
It is a lot. And I don't want to sugarcoat it. Like, it is a lot of hard work, and sometimes you're like, what am I doing? But if it's something you love and you're interested in your project, and I clearly was and had great mentors and supervisors, then you'll just like thrive. So, yeah.
Good.
It was good.
Excellent.
I think that's. I mean, she ends the entire session with compliments to you and me, so
Perfect.
I reckon we should probably stop there.
I think we should.
Thanks for watching.
Doesn't get any better.
Thank you.
Thank you.